Welcome back. In this screencast, we'll finish Chapter 3 by discussing the remaining organelles. Let's start with ribosomes. One way that ribosomes differ from other organelles is that they are not surrounded by their own membrane. They also differ in that they are found in both eukaryotic and prokaryotic cells. Ribosomes are a vital part of all cells because they are the site of protein synthesis. In eukaryotic cells, they're generally associated with a maze-like membrane extending out from the nuclear envelope called the rough endoplasmic reticulum. The endoplasmic reticulum, often abbreviated ER, extends through the cytoplasm from the nuclear envelope out towards the plasma membrane. The synthesis and transport of a variety of different organic molecules occurs in the ER. As mentioned a couple of slides ago, ribosomes are associated with the rough endoplasmic reticulum, which is found closest to the nucleus. It's the ribosomes that give the rough ER its rough or bumpy appearance. The synthesis of proteins and their transport to other regions of the cell occur in the rough ER. As we move farther from the nucleus, the ribosomes disappear from the ER. We are now in a region called the smooth endoplasmic reticulum. Picking just one function, the smooth ER is an important site of the synthesis, lipid, and transport of lipids. Here, the ribosomes are shown as red dots on the rough endoplasmic reticulum. The smooth ER is shown as being more tubular in nature. Both are more extensive than shown in this drawing. Front and center is our next organelle. To me, it looks like a messy stack of pancakes with butter and syrup dripping off the edges. This organelle is a Golgi apparatus, or sometimes called Golgi body. Botanists always have to be different. In plant cells, they're called dictyosomes. Golgi apparati receive, modify, and then package a wide variety of different kinds of organic molecules. They are most abundant in glands, the cells of which secrete a variety of different products. The small membrane-surrounded organelles that resemble drops of butter or syrup coming off the Golgi apparati are vesicles. They may contain a variety of different things, from raw materials to finished products or waste. In comparison, vacuoles are larger containers. Plant cells often contain a very large central vacuole. Lysosomes look like small vacuoles. They differ in that they contain digestive enzymes. These enzymes are proteins that speed up the rate of hydrolysis. Remember, this is the reaction that splits organic molecules into smaller components. When cell biologists were first trying to figure out the functions of various organelles, lysosomes drove them crazy, because if they're broken open, the enzymes contained within them digested the surrounding cell structures. In this animal cell, you can see small vesicles near the Golgi apparatus and a larger vacuole to the right. Lysosomes look similar to the vacuole. In this plant cell, the large central vacuole looks kind of like an oval balloon in the central part of the cell. Around the periphery of the cell, the green oval structures represent chloroplasts. Photosynthesis occurs in the chloroplasts of eukaryotic cells. They are found in plant tissues specialized for photosynthesis, primarily the leaves, and are also found in algae, the plant-like protistans. Pictured here, Spirogyra is a common alga that has a beautiful ribbon-like chloroplast spiraling up through the cell. Also pictured in this slide is blue-green algae, more closely related to the bacteria than they are to algae. It's prokaryotic and therefore does not have chloroplasts. Instead, photosynthesis occurs in the cytoplasm of these cells. Unlike most other organelles, chloroplasts are surrounded by two membranes. Inside of the organelle is a third set of membranes, which look similar to stacked poker chips. These are the thylakoid membranes, which collectively are called grana. The capture of sunlight energy used in photosynthesis occurs on the grana. Surrounding the grana is a clear liquid called the stroma. The second half of photosynthesis, when sugar is produced, occurs in the stroma. Chloroplasts are also different from many other organelles in that they contain some of their own DNA and ribosomes. I wish they'd been a little more careful when coloring this chloroplast. More accurately, only the grana should be green because this is where the chlorophyll is located, and the stroma should be clear. In some ways, mitochondria are similar to chloroplasts. Like chloroplasts, they have a double membrane and they contain their own DNA and ribosomes. However, the chemical reactions occurring in the mitochondria are just the opposite of photosynthesis. Aerobic respiration occurs in the mitochondria. This metabolic pathway breaks apart organic molecules, such as sugar, to release energy. Photosynthesis, in contrast, captures sunlight energy to make organic molecules. The outer membrane of mitochondria give it a sausage shape. The inner membrane is deeply folded. These folds are called cristae. The enzymes necessary for aerobic respiration are found on this inner membrane. Inside of that membrane is a fluid called the matrix. The endosymbiotic hypothesis offers an explanation for the origin of chloroplasts and mitochondria. It's thought that these two organelles were once free-living prokaryotic cells which were either captured by or invaded early eukaryotic cells. There are several lines of evidence supporting this theory. Chloroplasts and mitochondria are similar in size to bacteria. The outer membranes of both is no different from membranes found elsewhere in the cell. However, the inner membranes are more similar to those found in bacteria. The DNA and the ribosomes found in these organelles is also more similar to that found in bacteria. The cytoskeleton of a cell is much different from your skeleton. Instead of being composed of rigid bones, the cytoskeleton is made up of a dynamic network of fibers and tubules extending throughout the cytoplasm. In addition to providing structural support, the movement of the cytoskeleton moves the organelles and other materials around within the cell. In actively photosynthesizing cells, the cytoskeleton pulls the chloroplasts in a circular manner around the cell. The movement of an amoeba is the result of changes in the cytoskeleton. In lab, we saw cilia on the cells lining the human respiratory tract. In your lungs, the cilia sweep mucus and debris up and out of the lungs. They were also present on the surface of many of the animal-like protistans that you saw swimming quickly in the pond water samples. Cilia and flagella are similar in function, but they differ in size and number. Cilia are much shorter and more numerous. Flagella are often present in a single long tail-like structure, while in some organisms there may be 2 to 6. The green euglena here has a single flagellum, similar to that seen on sperm. Also pictured is a paramecium, which many of you saw in the pond water samples. It has cilia essentially covering its entire plasma membrane. This concludes Chapter 3 and the material that will be present on the first exam.